Hey Mustangs, in this video we're going to be taking a look at how prokaryotes regulate their genes. Okay, so we will be taking a look at prokaryote gene regulation. Okay, so prokaryote gene regulation, remember a gene is a section of DNA that has instructions for building a protein. So those genes can be turned on or off, and that's where the regulation part comes in. So regulating a gene means turning a gene on or off. Now, why would a prokaryote want to do that? Or any cell want to do that? So prokaryote, remember, is bacteria. So prokaryote is bacteria. And the bacteria wants to be able to turn its genes on or off. The reason for that comes down to energy use and materials use. The cell wants to be very efficient in these two things. It wants to be able to use its energy and material wisely. It doesn't want to waste energy. It doesn't want to waste materials. So genes, remember, have instructions for making proteins. So sometimes those proteins aren't needed. So if the proteins aren't needed, a gene needs to be turned off. If the gene's turned off, that's going to save energy and save materials because that protein won't be being made. If it's not needed, the protein doesn't need to be made. Now let's say the protein does need to be made. Then a gene can be turned on. And in the case when a gene is turned on, then it's an efficient use of energy and materials because it's making a protein that is needed at the time. Now this often is going to change. It's going to change a lot depending on the internal environment and the external environment of the cell. It's always changing. The inside and outside of the cell is always changing. Different things are happening inside, different things are happening outside of the cell. So different proteins are going to be needed at different times. Okay? So for regulation, it's turning genes on and turning genes off. That's what we mean by regulation. And prokaryotes want to be able to control that. They want to be able to turn genes on or off to efficiently use their energy and materials. They don't want to waste on proteins that aren't needed. Okay? Now, in order to really understand what's going on, real quickly, brief review of a metabolic pathway. So a metabolic pathway, remember, is a series of steps that includes enzymes. So here's enzyme 1, here's enzyme 2, and I'm going to stop just at enzyme 3. Some metabolic pathways have more enzymes that are involved in the process. But in a metabolic pathway, it's a series of steps that's going to change a molecule so that it can be used by the cell. Sometimes it involves modifying it in building a molecule in a certain way, or it can involve taking a molecule and breaking it down in a certain way. So in a metabolic pathway, you start with a certain molecule, not a protein. It could be a protein, but we'll just say it's some other molecule here. So this molecule is taken by enzyme 1, and it is changed a little bit. Then enzyme 2 will grab that molecule, and it'll change it, modify it a little bit. And then enzyme 3 will grab it, and since this is the last enzyme in this metabolic pathway, it changes it to the final product of what you are actually looking for. Okay? So this would be our final product in this metabolic pathway. Now, in order for this metabolic pathway to work, these enzymes must be present. Now, where did these enzymes come from? Gene regulation. Each of these enzymes has a gene in the DNA that has instructions for building it. So the DNA has the instructions on how to build enzyme 1. The gene within the DNA is transcribed into mRNA, then it's translated into the protein that you see here. Same thing for enzyme 2. It has instructions in the DNA that build it, and so on and so on. Now, these enzymes are very specific, and that's what the DNA instructions are. They say exactly how to make these enzymes. If these enzymes are not made correctly, this metabolic pathway shuts down. So let's say that there's a DNA mutation and in the gene for making enzyme 1. So the DNA that makes enzyme 1, the instructions get messed up somehow, and the enzyme isn't built correctly. Well, this whole metabolic pathway is going to shut down as a result of that. So metabolic pathways are really important because it makes products that the cell can use. So it either builds them or breaks them apart so that way the cell can, again, go back to this, use energy and materials efficiently. So let's go ahead and take a closer look at how prokaryotes regulate their genes. How do prokaryotes turn their genes on or off? In order to understand that, we have to take a look at operons. So first here's DNA. Okay. When you look at a bacteria, which is a prokaryote, it has one large circular piece of DNA that's all kind of wound up here. That's its chromosome. So that's a prokaryote chromosome. Picture a DNA double helix like this, where the ends come around and form a circle when they connect together. So it has a large circular chromosome. And then many bacteria have a lot of smaller pieces of DNA. Again, circular pieces. These are called plasmids. So you have the main chromosome, and then you have plasmids here. Okay. So plasmids are DNA that bacteria can pick up. So they can usually pick up these from the environment. When a bacteria picks up plasmids from other bacteria, that's called transformation. Okay, so anytime a bacteria picks up plasmids, transformation happens. So let's go ahead and take a look at how the prokaryotes are going to regulate the genes. So the key is operons. Okay. An operon is a unique piece of the chromosome here, and it's set up in a very special way because it's connected to metabolic pathways. So remember here, this was our metabolic pathway. And what happens is, in order to make sure that all of these enzymes are built all at once, the genes that encode for them are lined up one after the other. So if this was a section of DNA that has instructions for making this enzyme, and this enzyme, they would be right side by side. So this would be the gene for enzyme number 1, this would be the gene to make enzyme number 2, and this would be the gene to make enzyme number 3. Okay, so each of these is a separate gene. This one makes enzyme 1, this one enzyme 2, and this one enzyme 3. Okay. Another part of the operon is the promoter. That should sound familiar to you because the promoter is the region that RNA polymerase binds to in order to transcribe these genes. So remember, these are genes. These are sections of DNA that have instructions for building these enzymes. And if these enzymes aren't built, then this metabolic pathway can't happen. Another special part of the operon is the operator, which basically acts like the on-off switch. So we say gene regulation is about turning genes on or off. This is the on-off switch for these genes right here. Okay, so this, remember, is our promoter. Promoter region where the RNA polymerase binds. RNA polymerase transcribes these into mRNA. These are the genes, and then this here is the operator. In order to turn the operator on or off, there's another piece called a repressor. So a repressor is a molecule that can bind right here, and when it does bind, if this repressor binds right here on the operator, it turns it off. So think of the operator like an on-off switch. So picture an on-off switch like in your house. Okay, the on-off switch, if it's in the on position, that means that these genes are being transcribed and these enzymes are being made. If it's in the off position, and the way it gets put in the off position is if this binds to it, if it's in the off position, these genes do not get transcribed and these enzymes do not get made. Why would we want to do that? Efficient use of energy and materials. So if these enzymes don't need to be made because we don't need to make our product here, then this is going to bind to the operator, turn it off, and as a result, these genes are not transcribed. Okay. Now, each of these genes is going to be transcribed, remember, into RNA. So this gene here will be transcribed first into mRNA, then this one will be transcribed in mRNA. So each one gets transcribed in mRNA. Since they're one single piece, you can do something like this, okay? And they can be then translated later on, okay? Actually, let's go ahead and go back to the single pieces there, sorry. So each one can be transcribed and then translated on a ribosome. When they're translated, that's when the enzyme's made. So during translation, enzyme 1 gets made from there, enzyme 2 gets made, and enzyme 3 gets made. Okay? Then these can actually be used. Now, There's two types of operons. There's inducible and repressible.